8 hours. Did you know that?
Well, seems like something worth doing. It's just
That's why. So. Skinny hot. I thought when we moved from home, I was like, yeah. What?
That's your professional career or your personal? I'll call personal.
Your belt's really cool.
This one?
Yeah, it's like fabric.
This is like I wouldn't expect that.
Morona?
Oh, Marona's the brand. Marona's got to go on.
I'm not good at
I think
reading.
my sweater might be.
I'm just sitting on my computer so that if there's anything important, I can catch it. It's on your screen. I'm just. Good. Great.
These are my questions for this.
Won't have a satisfactory answer.
I hope so. I hope that's correct, because you need to be pushed to the next level.
You did that my first year of grad school. Are you, so you're still teaching conservation biology? You still do the groups with students?
Physiology. Physiology.
That was such a good experience. Yeah. He does discussion groups with grad students.
You know, I don't.
They discuss all the things.
Which is very good evolutionary things. Yeah. So, that's good.
Yeah. Yeah. I'm taking those. That your last one.
Yeah. Right. Yeah. Yeah.
Still big.
I'm not.
Yeah. Right. Yeah. I'm interested in like.
Yeah, and
Yeah. So it's yeah.
Katie was just going to say hi for a second. Fletcher, what's your TA? He always teases that I still
Whoever it is.
Yeah, he's here.
Okay, there it is. All right.
Hey Charles, I'm
Excellent. Okay. Yeah, so, well, joining us from New Zealand, we have Fletcher's committee member Rita Spilgalis, professor at Duke. So he's up at a truly ungodly hour, which we're grateful for. All right. Yes. Yeah, in the camera, hopefully, but out with the projector. All right. So, should be worried that I have notes. But, all right. So, I still clearly remember Fletcher's first email to me as a prospective grad student, specifically the first two of the first sentences. My broad research interest is in how community-level processes, such as interspecific interactions in multi-host parasite communities, drive population and community structure. I consider myself empirically minded and enjoy experimental field research. And I said, yes, that's great. And he turned out to be a very accurate and foresightful description. So at the time, Fletcher was working for a large environmental consulting company in the San Francisco Bay Area. And he obviously decided that pursuing a PhD would be more fulfilling for him. But he still wanted to contribute to environmental practice, especially in his native California. And so one project that he started was a large-scale survey of the meta-community of fungal pathogens in Northern California. The survey grew to be extensive enough, covering over 30 kilometers, that it got too big to complete all the sequencing in time for the defense. So that will not appear today. One major thing that he's almost done, but he has lots of other things to talk about today. Along the same lines, before graduating with a bachelor's in honors from UC Berkeley, he graduated from Santa Rosa Junior College. And after starting his PhD here at UNC, he was motivated to start an interinstitutional collaborative research project with faculty from the junior college involving undergraduates in junior college, which is a fairly unique opportunity for them, I think. And so that went on for several years. It involved up to a total of 15 undergraduates. So another thing he did on the side that was a substantial contribution that won't be on the screen today, I don't think. And let's see. All right, so in the lab group, Fletcher's been a very valued member, growing from an early mentee who was very receptive to input and feedback from more senior members to someone who now is a very active mentor in the lab group himself, which many others benefited from. He's always been highly independent, and his independence has benefited in part from his success in winning fellowship support. So he had several fellowships from within UNC and also an NSF graduate research fellowship. And let's see, so while Fletcher's now completing his PhD, I'm happy to say that he has agreed to stay on for an uncertain amount of time as a postdoc in the lab. This is great for all of us, and hopefully should be good for him. It's also very appropriate since our current grant in the lab builds very directly on data that he generated and ideas he generated. And so with that, I'll let Fletcher tell you about his data and his ideas.
Thank you, John. That was very nice. I thought that would be more damning than that. Can I turn off one of these lights?
So yeah, hit the arrow.
The one that says front row of lights. All right, I promised myself I wouldn't drink coffee before this meeting, and I did, so I'm just going to try and go slow. Also, there's a baby in the back of the room, and now you know what those sounds are. All right, so the general question that motivates my research is what controls parasite diversity in nature. And I study parasites that infect plant leaves, and I define parasite really broadly to include anything that spends one life history stage infecting a single host individual. So that can include things like fungal and bacterial diseases. It can also include some insect pests like leaf miting and galling insects. And specifically, I'm interested in what controls parasite diversity across scales. And today I'm going to talk about research that was conducted at 2 scales. The scale of plant leaves and the scale of whole plant communities. At the plant leaf scale, we often think of a host organ, like this leaf is a patch of habitat for maybe these 3 different colored parasites. And at the scale, we might expect interactions to take place as parasites compete for space or activate different immune responses in the host. And then just as multiple patches of habitat can be connected to form metapopulations, metacommunities, so too can these host habitat patches as we move from host organs to host individuals to host populations, all the way up to host communities. And at this largest scale of a host community, we might expect that characteristics of the host community that influence the dispersal of parasites from one host to another. And I just want to point out that even though this is an idea that was published as recently as last year, it's really a fundamental question that's been of interest to parasite ecologists for a really long time, since at least the 1980s. And what both of these papers, and to a large extent my dissertation, have aimed to do is to take existing ecological ideas and use them to better understand the ecology of parasites. So whether that's metacommunity theory or island biogeography. And I think this is an important goal for a few reasons. First, understanding the diversity of parasites within individual hosts might tell us something about host health. So in this study, they found that as the richness of parasites within a host increased, that's what's being shown on the x-axis, the chance that that host would survive declined. So in other words, hosts that harbor more parasite species were more likely to die from those infections. Host diversity across hosts in a community might be important for understanding ecological processes. So for example, in this study, Michelle Hirsch and colleagues linked the diversity of fungi in forest plots to processes that shape forest tree diversity in the southeast, like Jansen-Connell processes. And last, I think understanding parasite diversity is it's an important natural history question in its own right. By some estimates, parasites comprise nearly half of all species. And so it might be argued that if we want to understand ecology and biodiversity generally, then it might be worthwhile to think about the ecology and biodiversity of parasites. And furthermore, there's some urgency in understanding the processes that influence parasite diversity, as many of these species might be at risk of extinction in the next 50 years. Okay. My dissertation research takes this multi-scale approach to understand the diversity of parasites that affect plant leaves. Includes 4 experiments that were kind of broken up into 2 parts. The first part measured interactions among parasites and other organisms living on the plant symbiotically. I'm going to talk about one of the 2 chapters from this part today. And the second part examines ecological processes that operate at the scale of whole host communities. I'm going to talk about 2 chapters from that. All right, so the first project focuses on the scale of individual host leaves and asks how interactions among symbionts, including parasites and mutualists, influence their epidemics. And when I talk about interactions, I'm really thinking about any situation where the growth or establishment of a parasite or an individual is influenced by the presence or abundance of them. Right. Traditionally, these interactions were studied using what I'm going to call a baseline framework, where symbiont A always has the same effect on symbiont B in lab trials or longitudinal surveys. But ecologists are increasingly appreciating that even though there are interactions that occur in this way, an important subset of interactions called historically contingent interactions depend on the order or timing of past events. Maybe one of the most commonly studied types of historically contingent interaction is a priority effect, where the effect of species on each other depends on their sequence of arrival at a site, or maybe in the case of parasites and symbionts and a host individual. So for example, Adame Alvarez and colleagues were interested in how endophytes, those are symbiotic microbes that live in plants asymptomatically, how endophytes influence disease caused by the bacterium Pseudomonas. And they found that when they inoculated hosts with an endophyte before the bacterium, it reduced disease compared to a control without the endophyte. When they inoculated the host with the endophyte after the bacterium, it actually facilitated disease. It caused more disease. And while priority effects like this have been documented in many lab studies, we still sort of lack a good understanding of how or the degree to which they influence epidemics. And so with that in mind, we set out to address two questions. First, can we detect the impacts of within-host interactions in the field? And if we can, are they being influenced by priority effects? And to do that, we evaluated interactions among 4 co-occurring symbionts in the host graphs tall fescue in a series of longitudinal field experiments. Tall fescue is an herbaceous, it's a grass. It's been widely naturalized for the production of hay and turf. Across hosts, we manipulated exposure to the fungal endophyte, Epicloe sinopialum. Epicloe is a defensive mutualist. It's passed vertically from parent to offspring. I don't know if you can make it out, but these little squiggly lines are the fungal mycelium growing inside of the seed. And then we measured epidemics by 3 common parasites: Calototricum cereale, Coccinia coronata, and Rhizoctonia solana. And these parasites differ in their feeding strategies, and their growth rates, and their dispersal strategies. And so we expected that they might interact with each other in a variety of ways. And so we looked through the literature and we found evidence that we might see any mixture of any number of facilitative and antagonistic interactions if we went and looked for them in the field. And so this is a kind of complicated hypothetical interaction network. It represents the interactions that we thought might be possible to observe in the field. I'm showing facilitative interactions with these blue arrows and antagonistic interactions with red clubs. And really the main take-home message is that there are a lot of different ways these things can interact, and they can be either positive or negative. Okay, and to explore these interactions, we conducted a series of experiments. In each experiment, we grew plants in a greenhouse from seed, with or without that endophyte. We then embedded them as sentinel uninfected plants into a wild host population in North Carolina. And then we tracked infections by the 3 parasites. Okay, so the first question we wanted to address was whether we could measure the effect of within-host interactions on natural epidemics. And to do that, we used an existing modeling framework that was developed by Ian Fenton and colleagues to generate what I'm going to call the baseline model of interactions. And this measures biotic interactions among symbionts by testing how the abundance of symbionts in a host, that's the 3 parasites, or whether or not the host has the endophyte, how that influences the growth rate of some focal parasite. Following this framework, we tracked infections on individual leaves for the duration of the growing season, and then we modeled each parasite individually as a focal parasite and tested its growth as a function of the abundance of the other parasites in the endophyte. We did all of this using a series of longitudinal mixed models. All right, so what does our baseline interaction model tell us? Well, overall, we identified a mix of facilitation and antagonism like we expected. These dashed lines are interactions that we thought might be possible to see, but we didn't see evidence for them. And I want to take a second to note that even though we know the seeds had the endophyte, when we assayed the plants at the end of the experiment, we actually were unable to detect the endophyte, which means it was unable to grow out of the seed and colonize the plants. This was a problem with the seed. What's really interesting here is that despite the fact that the endophyte wasn't growing in the seeds, we still see 2 of the 3 interactions that we expected. I'm not going to talk about that more now, but it might be fodder for discussion. Okay, so the next question I wanted to ask was whether these within-host interactions are being influenced by historical contingency. And to do that, we modified the baseline framework. So instead of broadly testing how biotic interactions among symbionts influence their epidemics, our priority effects models test more specifically how the order of infection timing among symbionts influences parasite epidemics. So whereas this baseline model tests how symbiont abundance influenced a focal parasite's growth, the priority effect model instead tests how the risk of a leaf becoming infected by the focal parasite is affected by whether or not that leaf was previously infected by the 3 parasites, or whether or not the seeds came from endophyte-infected hosts. And we did this using a Cox mix model, which is a form of survival analysis. All right, I'm going to show you the result of that model. For reference, here's the baseline interaction model that I just showed you. I just simplified the names of the symbionts here. E is endophyte, Puccinia, Colototricum, and Rhizoctonia. I'm still showing positive effects with blue arrows and negative effects with red clubs. And here's a priority effect model. So right away, it's pretty apparent that these 2 modeling frameworks identify different mixes of antagonism and facilitation. Here's what one of those priority effects looks like. So in this figure, the black line represents the risk of a leaf becoming infected with Puccinia as it ages. Puccinia is the focal parasite in this model. And the red dashed line shows the risk of a leaf becoming infected if it's been previously infected with Colototricum. So picking an age, say 40 days, a healthy leaf has about a 50% chance of becoming infected with Puccinia. If that leaf had been previously infected by Colototricum, it only has about a 25% chance. So Colototricum is reducing the probability of infection with Puccinia. All right, so far I've been focused on interactions that take place within individual leaves. Now I want to consider these interactions in the context of a process that occurs across hosts in a population. And that process is parasite phenology. So in this system, parasites vary in their phenology. Colototricum tends to arrive first, right at the beginning of the growing season, followed by Rhizoctonia, which emerges sort of in the late summer, followed by Puccinia, which emerges towards the end of the growing season. And whereas a within-host priority effect happens when parasites vary in their sequence of arrival on a host individual, we began to wonder whether variation in parasite phenology could generate historical contingency among hosts in a population. In other words, does the timing of when parasites initiate their epidemics influence how they interact, and are there consequences for their epidemics? And to test that, we placed cohorts of sentinel plants into the field at 3 different time points. The first cohort went into the field in June. Colototricum was prevalent, but Rhizoctonia and Puccinia hadn't yet emerged. And all the results from those first models came from this cohort. The second cohort of plants into the field in July after the Rhizoctonia epidemic had begun, but before Puccinia showed up. So for these plants, Colototricum no longer had a phenological advantage over Rhizoctonia. And then sometime later, Puccinia had a chance to follow those. And then the third cohort went into the field in September after all 3 parasites were present, essentially removing the effect of phenology on symbiotic interactions. All right, this relatively simple manipulation had a pretty large effect on parasite epidemics. This figure shows the epidemic in the first cohort. The y-axis is the proportion of all the leaves in that population of hosts that's infected. So Colototricum showed up first, that's this red line, reached about 60% of leaves infected just as the Rhizoctonia epidemic began. Rhizoctonia is the green line here. Rhizoctonia peaked at about 50% and then started to taper off right as the Puccinia epidemic began. When we placed plants in the field after the Rhizoctonia epidemic began, we saw a completely different pattern. Rhizoctonia exhibited a massive epidemic. Colototricum's prevalence was reduced substantially, and Puccinia didn't even infect a single host. And in the third cohort, we again saw a different set of parasite epidemics. Right, what does this mean in the context of interactions among parasites on individual leaves? This model is the combined results of that baseline and priority effect models that I showed before. In the second cohort, we actually couldn't detect any interactions among parasites. We couldn't even include Puccinia in the models because there were no Puccinia infections. And we think this is being driven by this massive Rhizoctonia epidemic that actually killed many of the hosts, which is like the strongest priority effect you And in the third cohort, we see evidence for an interaction that we had hypothesized but hadn't seen evidence for before, and we don't see evidence for some of the interactions that we saw in the previous cohort. So this indicates that parasite phenology did influence biotic interactions among parasites. Okay, so what do these results mean more generally? These phenological differences had really profound impacts on parasite epidemics. The phenology of many species is shifting as the climate changes, and this really points to one mechanism by which climate change might alter disease dynamics in parasite diversity. Second, this measurement of parasite phenology is really capturing a population level phenomenon, and these models are capturing how that phenomenon affects interactions that take place on a very small local scale. And so this might be an example of a regional process influencing what happens at a local scale. And because parasites are so small and experience multiple generations per growing season, these regional processes can occur across small areas, across short periods of time, like a single growing season. And so this might point to a nice model system for testing experimentally regional processes. Okay, so far I've focused on parasites that infect the same host leaves on a single host species. As you recall from my original email to Charles, I was interested in multi-host assemblages. At this scale, I found that interactions can influence epidemics. In a chapter I'm not going to discuss, we also found that host immunity might be an important driver of these interactions in epidemics. And all of this occurs in the context of parasite phenology, which occurs across host populations. Next, I'm going to finally scale up from host leaves to whole host communities and ask how characteristics of host communities influence the diversity of parasites at this scale. So at the scale of a host community, I'm going to focus on two response measures, parasite richness and parasite abundance. Parasite richness is just the number of parasite species in a community of hosts. So in this simplified plant community, there are 5 different colored parasites representing parasite richness of 5. Parasite abundance is the total amount of parasites across all hosts in a community. Here I'm quantifying that using community load, which is the average amount of damage by parasites per unit leaf area in the pot. So it's really a measure of parasite density. All right, at this scale, 2 potentially important drivers of parasite diversity are host diversity and resource supply to hosts. And in the case of plants, we often think about resources like nitrogen and phosphorus. Theory indicates that increasing host diversity could increase or reduce parasite abundance. But most empirical studies have observed that parasite abundance declines with increasing host diversity. Parasite abundance is generally expected to increase with increasing resource supply to hosts. These drivers can interact with one another. So experimental fertilization actually weakened the negative effect of host diversity on parasite abundance in an alpine meadow system. I'm showing that with this arrow pointing to another arrow, indicating that resource supply is moderating this effect of host diversity on parasites. And there have been a bunch of studies exploring relationships between host diversity and resource supply on parasite richness. And it's generally expected that as host diversity increases, parasite richness will increase with it. And that as resource supply to hosts increases, parasite richness will decline. Again, we can put all these together, and we see a complicated system of relationships between host and parasite communities. And to explore some of these relationships, I established an experiment with 2 fellow grad students, Rob Heckman and Peter Wolfart, that aim to test the influences of plant diversity, soil fertilization, and natural enemies on a number of ecological processes. The experiment was conducted in an abandoned old field in Duke Forest. Just for reference, it's just adjacent to the experiment in Tall Fescue that I was talking about previously. In 2011, we established experimental plant communities at 2 levels of diversity. We planted 6 distinct monocultures and 6 distinct 5-species polycultures. So every polyculture represents the absence of one of these species from monoculture. We established 2 levels of soil fertility, ambient soil fertility, and an annual application of 10 grams per meter squared of nitrogen, phosphorus, and potassium. And then we maintained the diversity treatment for 1 year using landscaping fabric. That's what this black target is, and as well as hand weeding. Then in 2012, we removed the landscaping fabric, started the fertilization treatment, and after that, I went through and reported the abundance and diversity of parasites on all 6 host species. At this point, the relative abundance of some of these plant species has shifted, but only our planted species were present in plots. All right, and the first question I wanted to explore was just how host diversity and resource supply influence parasite resistance and abundance. Just as a reminder, we have 2 host diversity treatments, monocultures and polycultures, and then we established 2 levels of soil fertility. Ambient soil fertility, which I'll be showing with these black circles, and fertilization treatment, which I'm going to show with red. Okay, when we look at parasite richness, which I'm showing here on the y-axis, we saw that host diversity increased parasite richness, moving from left to right, but that effect was reduced in fertilized plots compared to parasite abundance, shown here on the y-axis, increased by about 16% as you move from monocultures to polycultures, and was unaffected by soil fertility. This positive effect of host diversity on parasite abundance is called an amplification effect. And even though theory tells us it's possible, it really hasn't received a lot of empirical support. And so the next thing I wanted to do was explore this a little bit more. And specifically, I wanted to test whether host richness, or the number of species planted in these plots, was actually driving this amplification. And the reason I wanted to look at that was that theory, I turn this thing off again, theory tells us that host diversity has really 2 components to it, at least in the context of the design of this experiment: host richness and host composition. And theory also tells us that as host richness increases, we should almost always expect to see a reduction in parasites. That's because as host diversity, or as host richness increases, the density of any host species is likely to decline, and that should make it harder for parasites to get from one host to another. In contrast, host composition can drive positive or negative relationship between host diversity and parasite abundance. That's because species differ in their traits, which leads some species to be more likely to become infected, better at spreading disease. I'm going to use this picture of an infected host to represent that kind of host. And those are called competent hosts. There are also host species that are less competent than their counterparts. And I'm going to show a less competent host with this uninfected grass. And as host diversity changes, composition might shift to favor more of one of these kinds of species, and that can drive this relationship between host diversity and parasite. And because of the way we designed our experiment, we expected compositional shifts to favor these more competent hosts. And that would lead to an increase in parasite abundance with host diversity, not because of host richness, not because of the number of species in the plots, but instead because of host composition. And this is through a process called the sampling effect of biodiversity. Because of the way we designed our experiment, polycultures were more likely by chance alone to have these competent species than the average monoculture. Okay, and so we hypothesized that we would not be able to attribute that positive effect of host diversity to host richness alone. And to test whether the effects of host diversity were being driven by changes in host richness, we modified our original model to include host composition as a random effect. This is a conservative test that allows us to ascribe differences to host richness only if the differences between a monoculture and a polyculture are greater than the differences among compositions within each level of monoculture. In other words, it tests for the effect of host species richness after accounting for composition. When we did that, when we accounted for composition, we saw that the effect of host diversity on parasite abundance became non-significant, indicating that the effect of host diversity on parasite abundance could not be attributed to host richness. In contrast, when we look at parasite richness, we still see a significant effect of host richness and its interaction with soil fertility. All right, so far we've seen that the effects of host diversity and resource supply might be influenced by characteristics of host communities. Next, I wanted to explore whether characteristics of parasite species might influence the effects of host diversity and resource supply. And in this system, parasites can be sort of easily grouped into two broad categories: microbial parasites like this rust fungus, and insect parasites whose larvae develop within a single host, like this leaf-mining fly. Microbial parasites, in this system at least, are generally passively transmitted by wind or rain splashes, whereas insect parasites are actively transmitted by free-living life stage, like this one with wings. Because microbial parasites are passively transmitted, they're expected to respond really strongly to host density. Density is reduced at higher host richness. That should lead to a negative effect of host richness on microbial parasites. In contrast, insect parasites don't predictably respond to the density of their hosts. And that's because they can actively seek out their hosts even at low host densities. As a result, we don't expect host richness to influence insect parasites. We still expected host composition to have that amplification effect on microbes and on insects, and that led us to two hypotheses. First, that host richness alone does not contribute to the abundance of insect parasites, so if we control for composition, we should see no effect. Second, that host richness could contribute to variation in microbial abundance, but this effect might be offset by the positive effect of composition. All right, we tested these hypotheses using a multivariate response regression, which allows us to compare treatment effects across multiple non-independent responses. All right, we found that insect abundance was nearly doubled in polycultures relative to monocultures. The y-axis here is showing a standardized response. It's the percent of the maximum insect abundance across all treatments. It's increasing from monoculture to polyculture. This is part of a much bigger set of results. And we saw no effect of host diversity on microbial abundance. All right. That significant effect of host diversity on insect abundance could not be attributed to host richness alone, as we predicted. And again, this is in contrast to parasite abundance. Both insect and microbial parasite richness, both insect and microbial richness were positively affected by the host richness treatment. This is what those results look like. Here, the standardized response is the percent of the maximum richness observed in each group. So they're both increasing from monocultures to polycultures, but microbial richness seemed to respond much more strongly than insect. All right, so in summary, we found that the effects of host diversity and resource supply on parasite richness and abundance depend on characteristics of host communities and characteristics of individual parasite species. And I think these results are exciting for a couple of reasons. First, disease risk is multivariate. It includes measurements of transmission and infection severity, mortality and colonization of new hosts, emergence of infectious diseases, but testing this multivariate construct has proven really challenging for disease ecologists. And what we did here was we used a multivariate framework that was developed for the study of biodiversity ecosystem multifunctionality, and it revealed some of these complex relationships between host diversity, resource supply to hosts, and the different components of disease risk. Second, these results might have implications for agricultural pest management and species conservation, which is really exciting to me because I do basic research for the most part. I grew up in the wine country, and so I'm just going to relate these to a personal experience of my own. Growing up, driving through Napa, I would see these signs all over the place warning people of the glassy-winged sharpshooter It's an insect that transmits a bacterial Xylella to grapes. And it's a classic case of a frequency-dependent parasite, just like the insects that I was talking about. And one way that vineyards try to prevent outbreaks of diseases like Xylella is to plant these diverse vegetative buffers around the vines, like this insectary at Benziger Winery. The results of this study show just how important it is to consider which plants are being planted in these kinds of vegetative buffers. If you go and choose a random group of native species like we did in our experiment, you actually might end up creating a situation where there's a higher risk of infection by diseases like Xylella. All right, one drawback to these models is that they represent a static snapshot in time. And that might be helpful for understanding parasites in constructed environments, but perhaps it's not as useful in more natural environments that change over time as host communities assemble. And so perhaps if we want to know how host diversity and resources influence parasite diversity in the long term or in more natural environments, it might be worthwhile to think about how initial host diversity and resources influence the composition or structure of some future host community, and how that in turn influences parasite richness. And furthermore, the characteristics of host communities can be quantified in many different ways. And how we quantify those might tell us something about the mechanisms connecting host and parasite communities. So for example, the relationship between the type of hosts in a community and parasite abundance might tell us something about the importance of host competence, whereas the relationship between the relatedness of hosts in a community and parasite abundance might tell us something about the importance of the phylogenetic signal and host range. And so this last chapter aimed to test this hypothesis. And we did that by running our experiment for 2 more years and allowing the experimental communities to be naturally colonized by the surrounding vegetation. Then in 2014, we recorded the identity and abundance of every plant species in every plot. And this time we recorded parasite abundance and richness on all species in a plot, totaling up to 80% cover of that plot, in addition to the 6 planted species. We then broke down the community into 3 characteristics. Host species richness is just the number of host species in a plot. Exotic host abundance is the relative abundance of exotic species in a plot, the only planted native species. And so this really represents the degree to which a plot is becoming invaded by exotic species. And then host phylogenetic diversity used a null modeling approach to measure the degree to which a plot was more or less phylogenetically diverse than random, given the number of species in the plot and their relative abundance. And we did that so that our measurement of phylogenetic diversity would be independent of our measurement of host species richness. Both of these measurements might be correlated with exotic host abundance, and that's what these double-headed arrows represent. And then we took this whole structure of relationships and we tested it using piecewise structural equation, which is a form of path. All right, so first I'm going to talk about host species richness, which was measured in 2014. Host richness was positively influenced by initial host diversity. I'm showing a positive effect with a blue arrow. This number is the standardized coefficient from the model. Host richness was negatively affected by resource supply to hosts. This is what these results look like. Monocultures are on the left, polycultures are on the right, and I'm still using black circles to represent ambient plots and red triangles for fertilized plots. These results are consistent with just a bunch of empirical studies in grassland. So not very surprising. As we predicted, host species richness was positively associated with parasite richness, so host species richness increased, parasite richness increased with it. And we saw no effect of host species richness on parasite abundance. So this is in contrast to that amplification effect that we saw when we constructed host communities at random. All right, next, looking at exotic host abundance. This mediator responded to our treatments, again, as we expected. Polycultures tended to have lower abundance of exotic hosts. Fertilized plots tended to have higher abundance of exotic hosts. We expected that as exotic host abundance increased, we'd see a reduction in parasite richness and abundance. That's because when exotic hosts enter a new range, they're often thought to leave their natural enemies behind. We did not find evidence to support that hypothesis. Instead, we found that exotic host abundance had no effect on parasite richness and actually had a positive effect on parasite abundance, leading us to believe that perhaps the exotic hosts in this system are actually more competent hosts for the diseases that we carry. Looking at host phylogenetic diversity, we saw that as host phylogenetic diversity increased, it led to a reduction in parasite abundance. And this supports a growing body of theory and empirical research as well. All right, so how do we interpret these results? Well, first, I think they highlight the need to consider the indirect effects by which host communities can influence disease. These paths are fully mediated. That means that after we include these indirect paths, there are no significant direct effects of our initial treatments on our measurements of parasite diversity in 2014. Second, the presence of these multiple indirect effects really highlights the need to consider the multiple mechanisms by which host communities can influence disease. You know, we didn't see a significant effect of host species richness on parasite abundance. We didn't see an effect of our initial treatment on parasite abundance. But when we look at the indirect effect of initial host diversity on parasite abundance, we actually see the dilution effect that we expected. It's just being mediated by changes in exotic host abundance and changes in host phylogenetic diversity over time. And then together, these results really highlight the need to consider the multidimensionality of the host community when we think about relationships between host communities. All right, so I was motivated by this question about what controls parasite diversity across scales. Just to recap, we started by looking at the scale of host leads. And at this scale, we found that interactions between parasites might be important for determining their epidemics. Scaling up, we found that processes that occurred across host populations, like parasite phenology, have the potential to strongly influence these interactions in epidemics. We then turned our attention to host communities and found that characteristics of host communities and parasite species play important roles for determining parasite diversity at this scale. And then scaling up again, we found that changes in host communities over time can strongly influence the diversity of parasites. And I want to just end by coming back to this idea that we should integrate existing ecological ideas to better understand parasite communities. In my first year of grad school, I participated in a discussion group with a number of fellow grad students, Peter White, and at the group, Peter posed this question. Is there a small set of fundamental laws or first-order principles that govern all ecological patterns and processes? This is a typical Peter White question. Or is ecology better viewed as a library of case studies where the outliers, contingencies, and other special circumstances define the natural world? And this question has really guided the way I've approached my research during grad school. And I think the results that I've presented here lie somewhere in between these two extremes. Yeah, we successfully applied general ecological frameworks to understand parasite diversities, but we really needed specific knowledge of the conditions in which they occur in order to really understand the results. And so this push to focus on the top might actually be doing a disservice to disease ecologists. And furthermore, over the past decade, as surveying and sequencing technologies improved, we're getting more accurate measurements of symbiont diversity and abundance in nature. We get estimates like 50% of the cells usually required microbial cells, or 40% of all described species of parasites, or culturable plant endophytes outnumber plant species by a ratio of 10 to 1. And this has really led to a renewed interest in trying to understand some of the processes that generate and maintain this diversity, which has reinforced this push to integrate what we know about symbionts into existing ecological ideas. And I'm hopeful that the work that I've presented here represents both a cautionary tale and a path forward. With that, I want to thank my collaborators. The Interactions project was conducted in collaboration with Charles Mitchell and James Umbenhauer. The plant community level projects were conducted with Charles, Rob Heckman, and Peter Wolfart. None of these projects would have happened without these people, especially Peter and Rob. This picture of them scowling at me is how I will forever remember my field time. I want to thank my committee for challenging me to think more critically about my research questions, how to address them properly. I want to thank my funding sources, as well as the Forest, the biology department here at UNC, especially Kenlon. I've been privileged to work with a lot of really amazing people while I've been here. Thank them. I will thank the Mitchell Lab for improving my writing, calling out my many half-baked ideas, helping me through mid-season fieldwork meltdowns, generally making my science that much better. I want to thank my family, especially my partner Simone. I want to thank my copy editor Ives, and I want to thank Charles, who's just been an incredible advocate for me. This is a picture of Charles presenting some of our research at a conference last summer. He's just been the perfect advisor. I can't imagine having a better mentor. And with that, I'll take questions.
Quick clarification question. When you started with your initial host diversity, you had 5 plant species, and they were all native, you mentioned. Did you find other native plants in growing into those same plots when you expanded to the host species richness? And if so, what category did you put them in? I mentioned it's not exotic host, but I'm just curious.
Yeah, so in another paper that Rob was the lead author on, we examined those responses of the plant community more directly. So there were 3 categories in that paper. Native residents, those were the species that we planted. The native colonizers, which were the species that we didn't plant to colonize the plot, many of those. And then the exotic colonizers. What I did in this was I measured, this is the relative abundance of exotic hosts. So it's just the relative cover of exotic species relative to all the native species, colonizers or not. In the experiment, Rob can correct me if I'm wrong, I don't think our treatments had strong effects on the abundance of the colonizers. It strongly reduced the native residents. Or, well, there were strong effects on the native residents and there were strong effects on the exotic residents. Rob gives me the thumbs up, so I didn't mind.
In the community cohort experiment, I forget the name, the 3 cohorts. It looked like the cohort was eventually confounded with time in season when a plant was added to that population. What do you think that interaction might be?
Yeah, it's interesting. So the cohorts of plants, they start at different times in the growing season. So there's that effect that we really can't discount. We do know that we track leaves over time, and so we're actually measuring interactions among parasites at time points that do overlap. And leaves are constantly emerging throughout the growing season. These plants produce about 1 leaf per year. And so we know that we're getting new leaves in these, that 3rd cohort of plants. We're getting new leaves coming up, but it doesn't matter because when they went into the field, that's its production. Moving forward, we're really excited to start thinking about how we can manipulate things like parasite technology that doesn't have that problem. So one project that we did this last summer, it's just wrapping up now, is we used fungicides on plots of extant vegetation to remove the parasites at different times. So we still have this time thing, but now at least the plants are all the same age, so they've been healed for the same amount of time. But it's really tricky. I mean, the best way to do it would be to inoculate plants with diseases. But we do these kinds of experiments because we think it's more realistic than inoculating plants with diseases, which has its own set of problems.
Yeah, really great talk. I'm thinking about how parasite richness and abundance might, well, I'll make it a question. Are those, do those have different effects on the success and fitness of the different hosts in a way that would alter how those hosts are then interacting and perhaps competing with, as new species are coming in life? In other words, you've done this in terms of the richness and the abundance of the parasites, but I'm wondering about the fitness consequences that for different hosts and how that.
Yeah, the feedbacks on hosts.
Yeah.
That's just, it's such a hard question, and I think people have devoted careers to trying to answer it. And I mean, with these, we're in a sort of simple model system here, but these are perennial hosts. So just testing that question can be really challenging. One thing that we did in this experiment that I didn't talk about was we had another factorial treatment where we removed fungi and insects with pesticides. And so that treatment is one way that we might be able to think a little bit about the effects that those pests have on the plant community. But because we're messing with the fungal and insect community so directly, it doesn't really make sense to measure parasite richness. Another way we can think about doing that is by creating these longitudinal analyses. So I have data from 2012 when we established the experiment, and data from 2013 that I haven't shown, data from 2014. And so we could think about constructing a model where we let parasite richness and abundance in 2014 or 2013 influence the composition of the host community in 2014. But those models get really tricky, they get really hard to interpret.
So maybe just, based on your experience with the system, do you think the effects of parasites on the hosts over this sort of 2-year time frame are enough that they would affect how likely exotic hosts are to invade, for example?
Yeah, definitely. I mean, we see effects of the pesticide treatments on things like colonization by exotic. Well, actually, did we see an effect of pesticide on exotics?
Yeah, 1 year.
1 year. But Rob's seen that in other studies as well. Fencing is another one that often in these short time periods can have pretty large effects. Tomorrow, Rob's going out and looking at the phenology pesticide experiment we did this summer to see if that influences the structure of the plant communities. It's a question that I'm really interested in. It's really hard, I think, to test. In general, I think in the literature when they talk about disease risk and like the effects of these things on hosts, parasite richness is sort of a surrogate for emerging infectious diseases. So if there are more parasites, there's a higher likelihood that one of them might be able to jump to the host that you're worried about. Whereas parasite abundance is this measurement of like actual infection severity and risk to the individual.
I agree, this fascinating study. And along the same lines about the questions about diversity and abundance, aside from the issue of how parasite abundance affects the second kind of step of the host abundance and so forth, there's also the question that you alluded to about whether diversity, just even in your immediate experiments 1 year, whether because the host diversity is related to the average abundance of the hosts also. I think we should include that possibility, which raises the question of whether the diversity of the hosts or just the average of abundance of the hosts, or the abundance of particular hosts, is actually the factor that determines or influences parasite abundance and whether they both do. Did you have any way you could
So in that,
separate the effects of
yeah, I
the
mean,
immediate effects of post-abundance and diversity on the parasites?
in that 2012 experiment, that's kind of what we tried to do. And we found that the richness itself, the number of species, if you're able to sort of isolate it from everything else, it doesn't have any effect.
So it sounds like it is then, in fact, the abundance of those.
So
The density
in
of
this situation, we can't, well, so with the design of this experiment, richness and density are pretty interchangeable. We planted these, it's either a monoculture at one density or a 5-species polyculture at the same density, which means every host is 5 times less densely packed. And what we find is that density doesn't matter at the scales that we're looking at. It could be that density doesn't matter because those effects are offset by these strong compositional effects. That's one possibility.
I thought you just said it was diversity.
It was richness. So richness and density are kind of interchangeable.
Right, you can't separate.
Yeah. What I think is driving these, and I think there's pretty good evidence for it, are characteristics of individual species. Some species in the system happen to be more prominent hosts, happen to be better at spreading disease, or tend to be more effective than others.
All right. So let's thank Fletcher first of all. So the committee will take just a few minutes in anticipation of a successful defense. I'm going to host a dinner celebration for Fletcher at Villa's starting around 5:30. Because it's getting late in the day, we're just going to adjourn straight there and enjoy something here in Cooper. But it's nearby, so I hope anyone who wishes can stop by and join us and congratulate Fletcher.
So that's an hour from now, right?
We'll be very quick on the committee, yes. 2 hours, 5:30.
I'll congratulate you.
Yeah, so the committee, let's be back in about 5 minutes. Great. Excellent plan. That should be getting it. Not serious. Get it out. That's not. That's
Okay.